And so more kind of tools and methodologies that we use for cancer. So again, earlier, sinabi ko na cancer cell proliferation is a hallmark of cancer. And so how do we study that in the lab? There's different methods. I'll share 3 with you today. So one is called an MTS assay. So MTS, it's a substrate that will change color pag na-metabolize siya ng cells na nabuhay. So not dead cells, but only live cells will metabolize this MTS. And when they metabolize, they form this formazine dye product. And that's what you see here. It's like a purple dye. And so what happens is, what you see is like the more cells you have, the more live cells you have, the darker it is. The less cells you have, the less cells you have, the lighter the color. So that kind of is a surrogate for cell proliferation. And then another is what we call the trypan blue assay. So trypan blue is a dye. You use to color, again, the live and the dead cells. So this is basically, pag buhay ang cell, they won't take in the dye because they have the cell membrane is intact. But when the cells are dead, they will take in the dye. So here you are, they're blue, because you lose the integrity of the cell membrane when you're a dying cell, and so that's how you kind of distinguish. You can kind of work out the proportion of live cells versus dead cells, and that's how you work out how much the cells are growing. And then finally, this is what we call the cell titer glow assay. So this is a bit more complex and slightly more expensive. It detects ATP. So I don't know if you've heard of ATP in your kind of science lessons and things like that. So this is kind of the energy currency of the cell. So it's adenosine triphosphate. And again, so this is a substrate that you add to a plate. And again, the more ATP the cell has, the more alive they are. The less ATP, the more dead they are, basically. And so again, you can kind of see the effect of the drugs on cells through these methods. And then what you do is, you do these cells, you plate them out, and then you read them, and then you can start analyzing the data. So this is an experiment that I did. So these are breast cancer cells treated with 2 types of drugs. So DMSO here is the control, and you can see here increasing doses of the drug from 0.25 micromolar. You increase the dose, you can see that the cells are dying. So again, this is from using a trypan blue assay, actually. So control cells very much alive, and as you treat the cells with the drug, slowly namamatay na sila. And you can graph this out, and this is how we present things in a paper or in a conference. So yeah. The next, I think, so we talked about how to detect cancer proliferation. The next thing is how we study cancer cell migration or invasion. This is what I mentioned earlier when cells acquire these legs and feet to move out and metastasize into distant organs. And so one method is what we call the wound scratch assay. So you have these monolayer, so single layer of cells in a Petri dish. And then you basically take a pipette and you scratch a line in the middle. And then you'll observe how they kind of migrate towards each other. And so then you can add a drug or something, anything you want, to see if it actually stops this wound healing process. And so, and that's kind of indicative na, it may actually suppress or inhibit metastasis. So this is very cheap. This is very easy. And you just need a light microscope. And so yeah, so this is something that I think this is well established. I think this was developed in the 80s. And so I think a lot of labs still use this, but people have moved as well to kind of more complex systems. This is what we call the transwell Boyden chamber. So essentially, it's a little well. You have the cells here on top, and then you have a membrane na maraming butas, and then you kind of add the media, the soup na I was talking about earlier, and then you allow the cells to migrate onto the other side of the membrane, and then you can actually count the number of cells here, and then to see, if you add a drug on top, does that inhibit migration of these cells towards the other side? So again, that's another system that we use to study cancer cell migration orientation. And these are the kind of data that you can get from these experiments. So this is from a Boyden chamber assay. And so you can see, you can stain these cells na nag-move sa ilalim ng membrane. So you can stain them and you can count them. I mean, by eye if you really want to, but you can use a computer to actually quantitate this. And so you can see this is just a cell line that you inhibit a specific protein, and you can quantitate this. And you can see here, this is the control at the top, and at the bottom you inhibit this protein called MECP2. And you can actually see, it's obvious that maraming cells sa control, and then here if you inhibit this protein, konti na lang. So konti na lang nagmamigrate from here to here because we inhibited this protein. So that's just an example of how we can kind of quantify this assay and show the effect of some drugs on cancer cell migration and invasion. Another technique we use is immunofluorescence microscopy. And so this is widely used to assess both localization. So saan where in the cell are the proteins are, and how much they are, how much they're being expressed. So here, I guess I said, this is, if you remember the picture I showed earlier, yung migrating cell. So this, how you do microscopy, again, you plate the cells on a cover slip, you add your treatment, and then you stain them with antibodies. So this is kind of the cross section nung slide. So you have the cells here, and then you add the antibodies, and the antibodies are actually conjugated or attached to this thing called a fluorophore. So this fluorophore, when excited by a laser, will emit a light, specific wavelength. They will emit, and that's when you see color, and you can visualize that in a microscope. So kind of really a technique that widely used now, moving away from light microscopy. People are using confocal, this immunofluorescence microscopy, to visualize cells. And so what kind of data do we get out of this? So this is something that I've done like years ago. I don't remember how long anymore. So these are breast cancer cells. These are the control cells. So imagine, if you remember earlier, I told you well-behaved, attached. So this is what they are. So this is FAC, the protein that I was very interested in. Here, very well on the side, nice and compact cells. And then we add this growth factor called heregulin. And you can see, I'm sure even if you don't know much about cancer cell migration, it's very obvious naman that you can see that cells start to separate from each other. And they start forming these focal adhesions, yung mga paa at mga kamay, and they start moving away from each other. And then I was working on this drug, PF878 from Pfizer at the time. And when you add this drug to this, you can see that it almost reversed that kind of process. So, dikit-dikit na naman sila, and they have this, let's say, kind of normal look. And so that's how you kind of use immunofluorescence to understand how drugs work and how they affect the protein and in cells. So that was good. And then you can take it the next level up as well. You can do multiple colors. It's rainbow. So I really enjoyed this part of research because I really enjoyed art as a kid. And when I realized I can do this, I was like, I'm just gonna do this all the time. Like it's like painting. So this one is, you can do multiple colors if you want to detect different kind of proteins. So you use different antibodies na may different fluorophores. So let's say this one is for the green color, and then the other one is for the red, green and red, the red color. So different proteins. And again, same thing here. I don't want to explain all the science behind it, but you can see this is the control cells, nice and separate yung two proteins na I was interested in, FAC and STAT3. And then when you add the growth factor again, heregulin, they start overlapping with each other. They become orange. So green plus red, they become orange. And then when you add the drug that I was working on, the PF878, they go back to being separate. So again, this was a clue on how this drug actually works on stopping that invasion of the cell, that migration invasion of the cell. So again, you can use this technology to understand the mechanism on how your drugs affect the cancer cells.